Chapter 16, Part 8, Antibody Immune Responses. Antibodies are used against exogenous pathogens and toxins, meaning the pathogens and the toxins, these are, for example, proteins that are made by bacteria. So, or structures of any kind made by bacteria. These are made by something else. That's exogenous. So with the T cells, this is stuff that's made by your cells. So this is the antibodies are going against things made by something that's not inside of your own cells. So we get these in response to specific pathogens. We can induce antibody immunity with T-dependent antibody and clonal selection. So this is antibody immunity that is dependent upon T helper cells to produce the response. So we have 4 steps: antigen presentation for T helper activation and cloning, differentiation of the helper T cells into helper 2 cells, activation of B cells, and then proliferation of B cells. So antigen presentation is to the T cell. Here's our antigen presenting cell. It finds the T cell that has the antigen binding site that recognizes the epitope on the MHC of the antigen presenting cell. So now this T cell is going to become activated and it's going to differentiate into a Th2 cell. That's the next step, the differentiation into the Th2 cell. Now this Th2 cell, it cruises around and it finds one of the B cells. It finds the B cell, if you have it, it finds it. It finds the B cell that recognizes that same epitope. And it stimulates that B cell and says, hey, one of my buddies over here. One of my buddies brought this to me. I thought you might be interested. And then the B cell, it goes through proliferation. All right, so this is how this is the T helper cells. Again, these T helper cells are the ones that get wiped out with HIV. So we don't get this crosstalk between the T cells and the B cells and the different kinds of immunity that they provide. So so with this functioning, we can get a T cell response to this pathogen, whatever it is that's making this epitope. We can also mount an antibody, a humoral response, by activating the B cells as well. Most of the B cells that are the cells that are made by proliferation are those plasma cells. I showed an image of those a few slides back. They basically just secrete antibodies that make that antigen binding site, and that's it. And they live only for a few days, and then their antibodies actually last longer than they do. And they can also release new, they can do mitosis and release more plasma cells. But the plasma cells themselves don't really last for more than a few days typically. For the B cells, the memory cells we get here, these are produced by B cell proliferation. They don't make antibodies though. They have the genes for it, and when they do produce cells that will make antibodies, the antibodies they will make will be the same as that original B cell. So they have the complementary B cell receptor, the same epitope that triggered their production will be what they respond to. And when that antigen is encountered, going through fluids, through a lymph node, then this memory cell will wake up and go through proliferation again. So the first time that you get an exposure to a pathogen, that's called the primary immune response, and we generally make a relatively small amount of antibodies are produced in this response, and it generally takes days before you get enough antibodies to be useful, and so it takes a while. It's not something that your body can do within a few hours. It takes days, often a week or more, to get any significant production of antibodies going. Remember, we start with a single B cell. A single B cell has the right antigen binding site, and it just by random chance happens to be there. If it's not there, then we just don't get an antibody response. Secondary immune response is when you get exposed the second time. So you've already got memory cells, and you probably have more than one memory cell. You probably start out with a few thousand memory cells, a few hundred at least, and several of those will get activated and go and do proliferation, and we get a much faster response rate, and we get more antibodies, and we get them faster by far. So this is what happens if we basically immunize someone for tetanus, give them the toxoid, wait, and we're measuring periodically, and we're testing, is there any antibody in the blood of the individual afterward. So we test them every so often, and then it's after day 3 before we even see any, and what we see here is IgM, and then we also start to see a little bit slower and less of it. Initially, we start to see IgG, and then the IgM sort of levels off and goes back down, and the IgG picks up, peaks at about 2 weeks, and then it goes down, and basically there are almost none after that. So that is our primary response. And this is part of why it takes a while. You get a cold or you get certain kinds of infections, and it takes a while before you get much response. And often by the time you do get antibodies, your symptoms are over anyway. But you're ready for the next time. And the next time that you get exposure again, we're going to get, by day 3, we've already got more IgG, basically almost as much as we got the whole time before, and we haven't peaked yet. And we also have a lot of IgM. And by day 6, we're almost done. We're on our way down. And the other thing is, look at how low the slope is on this. We've made a lot of antibodies, and they're going to persist for a long time. We've got more memory cells tucked away this time. If you've seen it once, you may or may not see it again. If you've seen it twice, you're probably going to see it again. So we tuck away more memory cells of the B cells and T cells, and prepare to see it again. And we'll have a fast, it'll be still very fast, very quick, and very high concentration of antibodies that come a lot faster. The next section will be the types of acquired immunity.